who is Shelly Krueger. She is a Florida Sea Grant agent in Monroe County for UF IFAS Extension. Shelly has a bachelor's degree from Georgia Tech, a master's degree from Savannah State University in marine science, and she is currently working on her PhD in sponge restoration aquaculture in the Fisheries and Aquatic Sciences program here at the University of Florida. And that's what she's going to talk to us about today. In 2019, Shelly wrote the Sponge Habitat Restoration module for the Florida Master Naturalist Program, Special Topics course in Marine Habitat Restoration. And as an extension agent, Shelly provides science-based education focused on water quality and Florida Keys natural resources. So thank you, Shelly, and I will turn it over to you.
How does that look?
It looks great.
Okay, perfect. Thank you, Laura. Today we are going to talk about what is sponge restoration aquaculture, water quality and ecosystem services, are the sponge propagules, also called the cuttings, reproducing in the FWC nurseries. The Florida Administrative Code defines restoration aquaculture is the cultivation of plants and animals to increase declining wild populations and improve the health of coastal ecosystems. For the Florida Department of Environmental Protection, for priority sponge restoration zones, the sponge density targets is 2,210 sponges per hectare. And sponge restoration is sorry, I have to move that so I can slide. Sponge restoration is a nature-based solution that improves water quality, biodiversity, and essential fish habitat. These are vase sponge propagules at a Florida Fish and Wildlife Conservation Commission nursery in the Florida Keys. Sponge restoration is similar to coral restoration in that the sponges and corals are both able to reproduce asexually through fragmentation. We cut fist-sized pieces of sponges, attach them to zip ties on the bricks to heal, and we place them into the nursery where they can later on be moved to a die-off area. This is a vase sponge 2 years later. You can see the sponge has completely reformed its vase morphology and looks identical to natural sponges. These are 4-year-old vase sponges, and the red arrow is pointing to other sponges that have naturally recruited to the area, and the bricks are no longer visible. Here are some other sponge propagules on the bricks in the FWC nurseries. A is a vase sponge, and you can see that the morphology is already returning to that vase sponge shape, and that the sponge is already growing over the zip tie. And B is another example of a restoration species. This is a yellow sponge. C is a glove sponge. And D is Cleonivarians, which we call elf sponge because it looks like the TV alien from the sitcom in the 1980s called ALF, for alien life form. And so we call these familiarly the elf sponge. And here are more examples of the shallow water species that are restoration candidates. Top left is a loggerhead sponge. In the middle, another vase sponge. Right, brown branching. Bottom left, golf ball. Bottom middle, that is a sheepswool sponge. And right, we have another glove sponge. And now we will transition into some of the water quality benefits and common ecosystem services that sponges provide. This is a map of the benthic habitats in the Florida Keys. When most people think of the Florida Keys, they think of Florida's coral reef, but in fact, the majority of the Florida Keys is dominated by seagrass and hard bottom habitats that support over 60 species of shallow water sponges. And these are highly productive ecosystems and are especially important nursery grounds for many commercially and recreationally important species. Sponges are filter feeders. They are feeding on phytoplankton, which is one of the smaller fractions of phytoplankton in the water column, like the size of cyanobacteria, also bacteria and viruses. They're essential habitat for spiny lobster, stone crab, and bonefish. And on an annual basis, three species bring in more than $150 million to the local economy. And that's through seafood, jobs, and the recreational backcountry guides is an incredibly lucrative activity that brings a lot of visitors from all around the world to fish for these species in the backcountry, which is part of why sponges are essential fish habitat. Sponges are also very important places for animals to hide and forage. Sponges have internal canal chambers that support an abundance of animals, including the only known marine eusocial species, the snapping shrimp Synalpheus species. This photo is magnified. In reality, the snapping shrimp, even a large one, is barely the size of my pinky nail. And they're also really important in acoustic signaling, and the noises they make can attract and recruit other fish and invertebrates to an area because it advertises that it's a healthy area for settlement and recruitment. So a lot of people don't know what a eusocial species is, but on land, an example of eusocial species are bees and termites. Because sponges cannot bite, sting, pinch, or swim away, they have evolved many chemical defenses within their tissues and the endosymbionts that live within their tissues. And so, for this reason, they are a source of many human drugs for cancer and viruses. And in Shelly et al., they say more novel bioactive compounds are obtained from members of this phylum each year than any other marine taxon. And if you look at the photo on the right of the volcano sponge, Tectotethia crypta, this was the first marine drug that has been used to derive drugs for AZT, herpes virus, and even more recently was involved in the vaccine for COVID. For this reason, they also have very few sponge predators. Some examples of predators are hawksbill turtles, starfish, butterflyfish, angelfish, trunkfish, cowfish, and nudibranchs. Since 1991, there have been a series of harmful algal blooms that have decimated sponge populations in Florida Bay. Unfortunately, the 2023 marine heat wave was devastating to sponges in the lower Keys. We documented a greater than 99% decline in sponges off Walt's Key Basin in Key West. And as you can see, these sponges, they're just dead in place. This is a glove sponge, dead in place. Another spongiid sponge, dead in place. But what's interesting is that, the Halimata species and the Penicillus, these macroalgae, there's some seagrass, even a little coral here appear unimpacted. And this is a photo of a loggerhead sponge before an algal bloom in Florida Bay and post algal bloom in Florida Bay. And the sponges just literally disintegrate in place in a matter of weeks during these blooms. Same with a vase sponge pre-bloom, a vase sponge post-algal bloom, and these are courtesy of Dr. Mark Butler from Old Dominion, well, formerly of Old Dominion University up in Florida Bay. While we do not know the reason for the 2023 die-offs in the lower Keys, the Florida Bay die-offs have been associated with Synechococcus blooms. And Synechococcus is a cyanobacteria. It's really responsible for making the formerly gin clear water in Florida Bay this pea soup color that you can see in the photo on the right. And one thing that's kind of interesting is that loggerhead sponges feed on Synechococcus. So this is an example of, you could almost say, the prey is killing its own predator. Unfortunately, this can create a trophic cascade, which can become a vicious cycle that is difficult to return to the previous seagrass and sponge-dominated ecosystems. You have a Synechococcus bloom comes in. It can lead to seagrass die-off because of the light attenuation. The water clarity becomes so turbid and cloudy that the seagrasses can no longer photosynthesize. view of a sponge die-off because of low dissolved oxygen. And then that in turn can lead to this vicious cycle where you have a decrease in water quality and clarity. You have a decrease in spiny lobster and stone crab because there's nowhere to hide and forage. And so this is what we mean by the idea of a trophic cascade. And so now I'm going to transition into one of my research questions is, are the sponge propagules reproducing in the Florida Fish and Wildlife Conservation Commission nurseries? And the good news is that thankfully, yes, they are. My objectives were to determine spawning months in natural sheepswool sponge, Hippospongiolacne, and loggerhead sponge, Speciospongiovesperium, using histology and light microscopy. Secondly was to survey gametogenesis following asexual propagation for 4 spongiid species, and again, the loggerhead sponge at FWC sponge nurseries. And one thing you may not know is that sponges are actually male or female. There are instances of hermaphroditism, but usually it's sequential and not at the same time. Although I did observe all my slides to see, and all of them were either female sponges in oogenesis or male in spermatogenesis. On the left is a sheepswool sponge. This is a ciliated pre-larva. These sponges, sheepswool sponges in particular, are internal brooders. So the male, this is a male spermatic cyst. Inside are these individual spermatozoa. If you see all these dark purple heads with the flagella, these are all individual spermatozoa. And these cysts are throughout the sponge. So speaking of, these are released into the water column, drawn into the sponge, fertilized, and then these well, this is a pre-larva, but when they become larvae, they are released free swimming as larvae. And then these are eggs in different asynchronous stages of reproduction. Because histology is expensive, we spent the first year seeing what time of the year the native natural sponges were reproducing. And we surveyed sheepswool and loggerhead sponges each month for a year. Here on the y-axis is 0 to 100%. On the x is May through April. And these are all the natural sponges not in a nursery. Okay. So what I want to point out is that the dark blue is oogenesis is occurring, the darker teal is spermatogenesis, and this lighter color means I found no evidence of gametogenesis in the samples I was looking at. And we found that the highest rates of reproduction were in April and May. I looked at over 250 histology samples of loggerhead sponges, and I only found one sponge that had eggs. And so this is a picture of that individual loggerhead sponge that had eggs in May of 2023. So for year 2, we actually sampled sheepswool sponge, glove sponge, grass sponge, and yellow sponge in the FWC nurseries. Since we identified April and May as the months with the highest rates of reproduction, during the second year, we sampled the propagules inside the FWC nurseries from April to June. And once again, 0 to 100% across the bottom is all the species combined for April through June. And so the dark blue is oogenesis. These are individuals with eggs. The dark teal is spermatogenesis, 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 spermatogenesis. And once again, these are all oogenesis. And what we found was out of 95 individual samples, 90% of them were in gametogenesis. And also it was highly skewed for males. 67% were males and 23% female. And the restored sponges had higher rates of gametogenesis and higher fecundity too, compared to the natural sponges that were sampled the year before. So on the left, this is a yellow sponge with eggs, all asynchronous in different stages. These are the fertilized embryos about that are getting close to be able to release and free swimming. And then you can just see every single one of these is a spermatic cyst. So, I mean, they were just incredibly fecund. It was nice to see. So if you want to learn more about what we talked about today, as mentioned at the introduction, the Florida Master Naturalist Program has a special topics marine habitat restoration. And of those modules, there is a sponge habitat restoration module, and also seagrass restoration, coral reef restoration, and artificial reefs. Shallow water sponges associated with seagrasses are critically important for water quality, clarity, and essential fish habitat for juvenile spiny lobster, stone crabs, and bonefish. Restoration aquaculture has the potential to re-establish sponges where they have been extirpated due to hurricanes, algal blooms, and marine heat waves. And I would like to thank the Environmental Protection Agency Region 4 Water Division for funding this research, the Florida Fish and Wildlife Conservation Commission for all their help allowing me to use their nurseries to sample their propagules to see if they were reproducing, Dr. Josh Patterson, Shirley Pomponi, and Christina Diaz, the people who helped me sampling on the boat and their expertise. I could have not done this project without Mimi and Simon Stafford and Doug Gregory. And all research performed under this presentation was all permitted by the Florida Keys National Marine Sanctuary permit. And that concludes my program. Oh, Laura, I can't hear you.
That's why. Thank you so much, Shelly. That was a great presentation and lots of cool stuff. We already have one hand raised, so Savannah, go ahead, and other folks, raise your hand, or you could also put your questions into the chat.
Yeah, thanks, Shelly. So my question is about your natural survey of the reproductive effort, and I noticed that it seemed like the peak egg production effort was offset from the peak sperm production effort. I mean, obviously the months you chose had the highest overall, but I mean, I don't know if you want to go back to that slide, but I was wondering if you had any thoughts on why the reproductive effort or gametogenesis or however you describe it would not be the same. Yeah, so like December and January looked like they actually had the highest egg oogenesis.
Yeah, it's highly skewed. So these are the natural sponges, and this is the propagules in the nursery. The dark teal is spermatogenesis. I don't remember the proportion, but I mean you can see it's dramatically higher for males than females. And they are broadcasting the spermatozoa into the water column, which means you do need them to reach the female sponges. But actually, in most of the literature, it's skewed for oogenesis. So I haven't looked into that. There are like difference in abiotic factors looking at coral reef sponges, which tend to be different species, and these near shore sponges that are in basically, chest deep water, that can have to do with like temperature and time of year, but I have not looked into that at all other than to note that that is what we found.
Thanks.
There's kind of a related question in the chat. Why do you think there was so little gametogenesis in loggerhead sponges?
So I am hypothesizing that they are similar to the giant barrel sponge, Zestospongia muta, which is found in coral reef associated environments and water more around like 60 feet-ish or so. So a coral reef associated sponge, but anyway, they are synchronized, infrequent broadcast spawners. So I'm thinking maybe they have some sort of a synchronized event that we just missed. Also, light microscopy has its limits. I think maybe I couldn't see the spermatogenesis using light microscopy. But all in all, I suspect there's some sort of a synchronized event occurring that we just missed. Yeah, because we looked in the beginning, we were just grabbing cores, like yay big. And then we started collecting the core from the surface all the way to the bottom because they are very large sponges and sponges don't have gonads. So technically, the reproduction can be occurring anywhere in that animal. So we started doing full cores. And even so, and there was activity, but all of the activity was always still at that top core. So we don't really feel like we were missing it based on early sampling of just doing more superficial collecting.
Go ahead, Carrie. You're welcome to unmute and ask your question.
Hi, so thank you so much for your talk. I was wondering what the next steps are for this program. Are you looking at potentially doing land-based assisted sexual reproduction with this information, or where does this research lead to?
There are groups at Florida Atlantic University that have been collecting larvae from sponges and then bringing it to land-based facilities to grow it out with the eventual plan to move it back into the Florida Keys. I haven't been involved in that research, but that is occurring. And so for us, really, the good news is that if you're creating these sponge nurseries and you're restoring die-off sites, it's really important that these animals are reproductive and becoming self-sustaining. And so we've seen that at the coral nurseries with the coral frags. I might be quoting wrong, and I'm sure there's someone out here who knows the answer. But I think if they're becoming reproductive by year 7, that's considered success. And so most of these animals were 3 to 6 years and they're highly fecund and they are reproducing, and size didn't seem to be a factor. I mean, some of these were still fist size and they were highly reproductive. So from a restoration standpoint, it's really good news that they're able to recruit to the surrounding area.
Awesome, thank you.
I had a question sort of um, about that. You said that your sponges in your nurseries had higher fecundity than the natural sponges. Why do you think that is? Are they stressed, or is it like is the nursery a really good habitat? Like, why do you think that is?
That is a really good question. Uh, I don't know because I mean, I literally found a sponge that I had propagated and I knew it was 4 years old. And I mean, it was the size of my pinky finger. And it was a male that had so many spermatic cysts that I could see it on 4 times my magnification. And I don't know. I just can't answer that question.
Yeah, yeah, okay. Um, there's not another one, so I'm going to ask a second question. You talked about a lot of different sponges and how important sponges were in general for their ecosystem functions and services. And I wondered if you could, one, say if like the ecosystem functions and services are similar across sponge species, or are there some more important than others? And similarly, like, is it easier to propagate some sponges than other species?
So to your first question, there are HMA sponges, which are high microbial abundance sponges, and all of these sponges are high microbial abundance sponges. There are also low microbial abundance sponges. And so really the difference is that the HMAs have like 3 to 4 times magnitude higher abundance of symbiotic bacteria and viruses inside their body tissues versus low microbial abundance, it's about equal with seawater. And what's interesting that we found after the die-off in 2023 was the 3 survivors, which was golf ball sponge, volcano sponge, and cleonovarians, are all LMAs. They're all low microbial abundance. They have higher pumping rates. They obtain more of their food from physically eating the picoplankton that they're pulling in and consuming versus the HMAs. There's more interaction with their endosymbionts to create the things they need for their growth and metabolism and reproduction. So that was kind of an interesting difference was the LMAs were like the survivors in this marine heat wave scenario. And then your second question, you're going to have to remind me.
It was you you mentioned like the propagation techniques for sponges. Are they the same for all of the species, or are there some species that are easier to propagate than others?
It is the same for all species. Basically, a this size part zip tied onto a brick left in place to heal over. Um, we've It's interesting because I've talked to people in counties, I believe it was Hernando, and yellow Spongia barbara was their least successful. Spongia barbara is our best.
Mm.
So yeah, there's definitely differences. Glove sponge does really well. We've heard in Marathon that sometimes they haven't had great success with Spongia graminia. So yeah, it is really interesting. And then loggerheads do tend to shrink up, and so sometimes they can float off their zip tie. So it's important to cinch them really tight, and then FWC might put some sort of like a orange bag over them just short term so that they don't float away. Um, and then fire sponge. They don't do fire sponge anymore. I heard that it didn't do so well, and also no one on the boat wants to deal with fire sponge because it's like getting a minor acid burn.
Fair enough. And if folks are interested in that Hernando project, there is a Nature Coast Currents on that project in our archives. We do have another question in the chat. Are there future plans for sponges in the nursery to be outplanted, and if so, where?
So the FWC in Marathon has a project right now to restore 30,000 sponges. And so they're currently um, getting those already in their nurseries. I don't know where they plan to outplant them, but they actually have a pretty robust plan of to put them in areas that haven't been perennially impacted by algal blooms. But I think we'd have to ask FWC about their future plans, but there are plans.
Okay, cool. Um, there's another question popping up in the chat. Is there a succession of sponges as a disturbed habitat approaches climax?
So anecdotally, we have seen that glove sponge, Spongia graminia, yellow sponge, Spongia barbara, those two seem to be your first like successional species, just based on the die-off we've seen at our nursery site. So yeah, oh, and also base sponge tends to be weedy. You have your like weedier, more um, quicker to recruit species, and then your loggerheads and things like that. We wanted to do a study for a long time to see how old some of these loggerhead sponges are because they grow so slow and they're so large. Um, but as far as I know, there have not been studies on like the succession of what returns following a die-off.
Thanks. Anybody else have questions? Please type in the chat or raise your hand. But if not, I have one more. Are there people who are using these techniques to grow sponges for harvest, or is it primarily for ecological restoration?
Elsewhere in the world, elsewhere in the world, they tend to suspend them and tie them to rope. And they have been used in like polyculture situations where maybe you're growing abalone for like aquaculture and abalone for consumption, and they may have sponges uh, tied from ropes around them to kind of help remediate some of the waste products and such like that. Um, the brick technique seems to only be used that I know of in the United States for restoration.
Cool. There is another question that popped up, and that is, I know you mentioned that the sponges you outplanted survived very well, but that some float off of the brick. What are the typical success rates of outplanting?
We just planted 1,138 bricks, and our mortality rate was 4.6% as of last November. So that was 53 sponges that we found empty bricks. And so we're hoping we are going to go out in a couple weeks and survey that nursery again and see what we have. But um, the mortality is in our case has been less than 10% over the last since 2017.
That's crazy high. That's cool. Um, One last question, and it followed up on a question I asked you. They're wondering if the difference in sponge reproduction between the wild and nursery groups could be related to differences in water quality, for example, silica availability or other nutrients.
Yeah, the propagules are in deeper water with very good flow, and sponges are density dependent in terms of what they're feeding on. If you have 100 people at the buffet versus one person at the buffet, that one person is going to get more food. So that could be some of the differences of, less energy. In general, sponges can use the ambient currents in order to conserve energy on pumping. So it could be that deeper water with better flow, and maybe there's more food there we haven't looked, but there's definitely, differences by location for sure.
Cool. Well, thank you. It looks like our questions are slowing down. That was a great talk and some great discussion afterwards. I really appreciate it. And I hope that folks will join us for our next webinar, which is May 13th. And Robbie Lamb will be presenting on nursery habitat for juvenile sport fish. So thanks, Shelly. Thanks everyone else for coming, and hope to see you guys next month.